items of this list depending upon how turbo we can pass through. I took a look at chapter 24 in preparation for today, and a lot of it is kind of like miscellaneous information that you already know about, so we don't necessarily need to review stuff that we've already reviewed. We're going to talk about the origin of life and the movement of life to land, just very briefly, and then we're going to move on to 42. If we do add an item, the item that we'll be adding is cellular respiration. So we did a photosynthesis review last time, cellular respiration, getting a little opposite in to do a review. And that'll be a fast, working, chanting, turbo chapter. So we'll be just shoving that right down your throats in a nice way. Okay, so the first thing that I want to do is I want to go over some of the worksheets that you guys have. First things first. Let's look at where it says Appendix B, AP Biology Equations and Formulas. I got this from the website. This is what I was trying to pull up with the no Wi-Fi in the multi-purpose room when we were together. This equation and formula sheet is the exact equation and formula sheet that you will receive on the test. So this is yours. It's fantastic. It's only as good as the user. What do I mean by that? Know it. Know how to use it and know what's on it. Just looking at a couple of things here. First of all, if you look at the first one, mean, that makes me kind of go crazy. Instead, I could just add up all the numbers and divide by the what? Total number. That's how to find the mean. It's also called the average. So equations that look like that, just go ahead and like cross them out. Because they do a little bit more than help. They kind of overwhelm you. We'll be doing some sample questions from a packet that I outlined, that I copied out for you, and we'll be looking at those. The other problems that are shown on here are the example problems that College Board gives you. They have the science practices and the learning objective. To you, this should be relatively pointless right now in your studying. We've also added a couple of other questions throughout this. But this is what you will see on your grid-in portion or on your math portion. Put that aside for right now, but you will be referring back to that as a formula sheet, so don't put it too far away. Talking about the same thing, the AP Biology exam practice grid-in questions, number 1. See, it has a little number 1 in the top right-hand corner. If you flip through that, those are the ones that you're going to be doing on your own. But if you see the little grids, that's how we'll be answering questions. One of the things that I want you to put a little star on at the front of this paper is I want you to say, read the question. If it asks for you to give the answer in the hundredths place, you better put it in the hundredths place. In the tens place, you better put it there. If it asks as a whole number, what are you going to do with that number? You're going to round it and you're going to bubble in the whole number. So numbers 1, 2, 3, 4, 5, you're going to be working on together. Now flip it, open the packet up. I put a little 2 in the right-hand corner. It says grid in practice questions. We're going to be going over these together. So you can keep this with your formula and equation sheet. That will be next. Those are the 2 things we're going to be working on together. The next thing that you see here, and that we'll be working on, I want to say 5 or 6 of them together. Then I'm going to give you some time to do. Am I too loud? Can you? Okay, good. Then I'm going to give you 3 to do on your own. And once you get time to do them, we're going to grade them in here and see how well you do. And then hopefully you'll feel a lot more confident about some of these things that you're seeing on the formula sheet that could be giving you relative anxiety. Now, the next one, please pick up the AP Bio Graphing Practice Packet. Excuse me. I pulled this off a website because every year I've taught AP Bio for the last 6 years, there's been a question that you had to graph something. What I don't want, it's embarrassing to you and it's a little bit embarrassing to me. If the question has to do with change over time, you definitely don't want to throw up a bar graph or a box and whisker or something like that. What is change over time? It's a line graph. So I have a little PowerPoint that we're going to go through which graph to use when. And it also comes from College Board, so it kind of helps with that. You can stick that away for right now. We may do one graphing practice together. Okay, the next thing I want you to pull out is a staple packet, 2013 AP Bio Free Response Questions. So actually, in a very smart way, thought that this would be a good idea for you to see how the packet will come to you. This is exactly what it will look like. Obviously not on Xerox paper with a staple in the left-hand corner. It'll be appropriate testing materials. But these are all of the questions that they got last year. What do you notice about the very last question? You did it. You wrote on this one. So it separates your short answer questions from your long answer questions. And it's a good idea as to how that last 50% of your test, which you might want to write in the top right-hand corner of this. This is worth 50% of your test will be factored in. So a couple of those sample tests that you've been given already. You've been given number 1, you've been given 2, and then hopefully you also have one in your review books that you've been doing. If you calculate through your multiple choice and find a percentage, and then you were to grade yourself on this, because on the College Board website is the FRQ key that tells you how you would earn each of these points. My suggestion to you is you write on these. You write these and see how well you would do with them. Also, going back to your review book, write on them. They also come with key cards, so you know where you're going to earn your points. Grade yourself tough. Grade yourself like a grader would grade you, because you don't want to go in thinking, hey, I got 100% on that 50% part of my test, and then how could I possibly have gotten a 20%? My goodness. So grade tough, and then when you leave the test, you'll feel more confident knowing that the readers really don't want you to fail. They really want you to do well. So they're going to grapple for you to have points, but only if you're organized. The next one, 2005 AP Bio Free Response question. That goes with this paper. It's a lined paper, so you can stick those together. We will, time permitting, have this as item number 6 to walk through after I talk about cellular respiration. The last thing that we copied for you is this major human endocrine glands and source of hormones. This is going to come from the hormone chapter that we're going over today. Well, no, that was already gone over. Yes. So this is the pituitary gland, what it does, parathyroid, pancreas, and important points are underlined. I would love for you to know this. Hormones are really important in the functioning of the human body. They allow us to stabilize our internal environment, something we like to call homeostasis, aka balance, maintaining balance. So I would know how these hormones do that, and I would commit this to memory / learn it. All right, without further ado, let's get into our math review. That rhymes. Okay, take a look at your grid in practice number 2. I'm going to make my way over to the Elmo. And we're going to do some of these together.
This is so funny. Maddie, this is so funny. Maddie, I'm zooming in. Maddie, I'm zooming in so that you can see the board better.
Okay.
Oh, shit. What do I want to say?
Oh, here's an example of something. This is the calculator that you are allowed to bring on the AP test. This is considered a 4-function calculator. You should not be bringing anything else. They also will have no replacement calculators for you. So if you come ill-prepared, that's it. You're solving your square roots on your own. So this is what you need to bring. You can see that it has a log, it has pi, which is going to be necessary. It has your square root, and it has your squared button. Make your life a little bit easier. I brought a couple somewhere. I don't know where they are. I was going to lend them out. Oh. Here, pass them around. Anyone that needs calculators. If you already have a calculator that you can work with today, don't take one. If you need one, they're coming around, but I want them back, please. Okay, so let's look at this. Well, I wrote you'll get this on the AP test. Okay. Grid in practice questions. Now, the reason I know that some of you guys in the back aren't able to see this, but you should have it in front of you. So we're on page 2, and it says these different directions. Use a 4-function calculator in the AP Biology equations and formula sheet that I have right here next to me while I'm working. This, I believe, is perforated on your test, so you'll be able to rip this out of your test, and you'll be able to work through the problems with it as you travel through it. Grid in each final answer from left to right. Don't round until the end. Then round your answer according to the instructions provided in the question. Example, round to the nearest tenth. So this is the first question that we're getting here, and it happens to be a chi-square test. Now this is something that we haven't done in class yet, right? So one of the things that you need to know, and if we actually hadn't done, if we hadn't reviewed on this, you would see it in your formula sheet, but you can see that there's a chi-square. It tells you right there. So one of the things that I want you to get in the habit of doing with these grading questions is I want you to start underlining what it's asking you to find. It's asking you to find a chi-square. That's the equation you're going to look up on here. Worst case scenario, you get partial points on it. You know what I mean? You work out part of the question. So this is your chi-square analysis. I'm going to look on my formula sheet and I'm going to find the chi-square value. That equation, that equation is the sum of O minus E squared over E. What is O? Observed. O equals observed. And then E is expected. Raise your hand if you're in stats. Awesome. Now, we are also going to use something called a degrees of freedom table. The degrees of freedom is just underneath where it says the chi-square test, or the chi-square. It says chi-square table, degrees of freedom. It gives you a p-value, and then it gives you all of these different numbers. Put a little star next to 0.05. That's the one that we're using. We're going to accept anything 95% and above. So if it's 95% validity, we're good on that. It doesn't need to be, what's the other one, assume? 99. It doesn't need to be at 99%. So you're going to either accept or, stats people, do we ever reject? No, we fail to accept. So the two things that I would write on my chi-square, you're either going to accept, you're going to reject or fail to reject. So you're going to reject or fail to reject. I'll explain that in just a minute. We're going to go through a problem that actually explains this. So next to your degree of freedom, it should say reject or fail to reject. So what are you doing when you're failing to reject something? You're technically accepting it. But you can't accept something if it's a probability. Does that make sense? The only time you can actually accept something is if it's 100%. Is it 1,000 degrees in here? Yeah. Okay, just checking. So, let's look at this first one that we have going on.
I zoomed into the
and then we'll walk through it for you. So first thing I want to do is, because I'm setting this up and I have a limited amount of time, but I looked it up on my formula sheet, I'm going to write my formula so I know exactly what I have. Equals observed minus expected squared over the expected. Then I'm going to read my problem. Eye color is sex linked. Oh dang, sex linked. That means it's either, that means, what is sex linked really? X-linked, baby. Sex-linked is X-linked. So now I'm concerned mostly with my X chromosomes. That's what I'm going to deal with. And I know that gender is something that's going to be considered. Drosophila melanogaster, we know all about that guy. Red eye color is dominant to white. A heterozygous female, so we know we're dealing with gender, so we're dealing with males and females. Let's write them out. We have a female, and we're going to have a male, because we're talking about mating for sexual reproduction purposes. So, we have a heterozygous female. If she's heterozygous and red is dominant to white, she's X what?
big R.
X big R, X little r. Was crossed with a red-eyed male. This is sex-linked. What is he? X?
Big R.
Big R. Why? Nothing, because sex-linked is X-linked. In the F1 generation, 134 flies had red eyes and 66 flies had white eyes. Remember, these numbers are quasi-important, but they're only important to give us statistics. They're only important to give us what is the ratio or what is the relative percentage of individuals that we have going on here. Calculate the chi-square for these results, round to the nearest hundred. So the first thing we're going to do is we're going to do this Punnett square, because it wants to know about the F1 generation. So we're going to cross this out. X big R, X little r, X big R, Y. Big, 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 little, big Y, little y. Okay, you got this? Now it wants to know the flies that have what? Calculate the red eye is dominant. So it wants to know, calculate the chi-square. So we're looking for red-eyed individuals and white-eyed individuals. Well, who in here is red eyes? What percent? 75%. So we can say 75 red, which would mean 25 white. If they wanted this in a ratio, by the way, this is percentage. They want this in a ratio, it's a what to what? It's a 3-to-1 ratio. 3-to-1 ratio going on here. All right, now that we have that information, we have our expected or our observed. We have kind of both, right? We have a little bit of both that's going on here. But this is showing us what really should be expected based on this information. We haven't considered these numbers yet. So this is going to be our E. This is our E, or our expected. So if we're putting this into our equation over here, because it's kind of a multi-step process, we don't know O yet, but we know our E is, what do you think? What do you think for red? What's the total here? What's 134 plus 66? It's a nice number. 200. Happiness and rainbows. So we have a total number of 200. What's 75% of 200? 150. So now you're going to say my E is 150. And I got to square that, and that's going to go in another place too, 150 down there. How are we doing so far? Okay? Okay, so then we're going to find out, we're calculating this for red first. We also have to do this for white, but we're calculating it for red. Now, we have, excuse me, so this is red. Now we have to find out our observed. So, what's our observed? There you go, 134. So if you were to do this, 134 minus 150, but then you square it over 150. Can somebody do that for me, please? Or maybe 0.71? 1.71 is what we get so far. Okay, so now we have this situation. Well, from here, we're going to have to do so this is red. I'm going to write. We're going to have to do our white. So let's carve out some space, like right here to do white. Okay? White, we have our same equation. Observed minus expected squared over expected. So, what is my observed for white? 66. Minus my expected for white? 50. Good. Because we're using these numbers, 200, but we're coming up with what percent of it? What? 25%. Is everyone with me? Let me do that again. We have a total number of flies, 134 plus 66, which adds up to 200 total flies. But we said 75% of them are red, meaning 150 of the 200 are red, and that would leave 25%, which will be white. So now we have, if we were to plug this into this appropriate number, it really should be over 50. Don't forget your squared, and then over your 50. What's your value? 66 minus 50 squared over 50. 5.12. Everyone get that? 5.12. So now we have these two values. What do we do with them, statisticians? We add them together, right? We're going to add these two things together. So here we're going to take 5.12 plus 1.71. Nice that they're together. What do they add to be? Thank you. 6.83. Then what? Now what are we doing? Okay, so now all it says is round to the nearest hundredth. Did we do that? Yeah. We're done. So can I put a 6 in, can I put a 6 in this first box? Yeah. No, because there's no 6 to bubble in. I'm going to say 6.83. And then I'm going to bubble those in. 6.83. Sasha.
Why don't we square root it because it's x squared? Like the formula?
Because we're looking for the chi-square value, and the chi-square value is x squared.
Okay.
Like that is the chi-square. We don't want to find x. Okay. To take this further, you would look at your degree of freedom, and you would find we generally are going to do, well, how do you figure out your degree of freedom? This is a pretty easy thing. You look at the number of phenotypes. So if you're taking notes related to this, you're going to look at the number of phenotypes, right? And then minus 1. Number of phenotypes minus 1. So what could these flies be? They could be red or they could be white. What is your degree of freedom? 1. So they could be red or they could be white. And if you count this, 1, 2, that's 2 minus 1, which is really 1. So you're looking at 1 degree of freedom. This is not part of the question, but I'm just showing you if you were to ask this, okay? So now you're going to look at the degree of freedom table, and your chi-square table, and you're going to look under 1. Then you're going to say, which one do we care about? 0.05 or 0.01? 0.05. So this is the one we're dealing with because we care that we can accept it, we can fail to reject it 95% of the time. And we look under degree of freedom 1, 3.84. As long as 3.84 is as long as our number that we receive is greater than 3.84, what are we going to do? Fail to reject. Yes? Okay, so what's our number? 6.83. Is it bigger than 3.84? So if we had to take this a step further, we're going to fail to reject. Now, this isn't going to be a graded question. How come? Because they're not words. It's numbers. But it could be a short answer. So I want you to understand how that works. You do need to know how to calculate your degree of freedom, to take the number of phenotypes that are given and you subtract it by 1 to figure that out. Okay, let's move on. Oh, by the way, if you're confused by this, look up here, guys. See all these? See all this up here? This tells you all of the size. Like N equals the size of your sample. O equals your observed. E equals your expected. Degree of freedom is equal to the number of distinct possible outcomes minus 1. Sample is standard deviation. So this tells you other things that are related to this. Do you mind if we table questions till after class? Because I want to push through. Is that okay? Okay, if you have questions about chi-square, you can talk to me or you can talk to Spo when you get into class mañana. All right, we're 20 minutes in. Gotta go. Next question. Next question looks like a situation that should be really easy. It should be easy to your 6th grade self, right? But then you realize you haven't talked about pi r squared in a minute. So you want to make sure that you know what's going on here. Well, it's asking you, what is surface area to volume ratio for the cell pictured below? Surface area to volume ratio. So I want this in a ratio at the end of the day. Or it's going to give me, we're going to round it to the nearest hundred, so it's going to give me a decimal. So, first thing, what did they give me? They gave me the radius. The very first thing I'm going to do is I'm going to look up on my sheet, and I'm going to try to find surface area or volume. Surface area or volume. Oh, I found it on the back side, on the bottom left. It even says surface area and volume. Now, I then have to decide what type of shape is this? It's a sphere. So I'm looking at sphere, and I have the volume of the sphere to equal, and I'm going to write that in here, V equals 4 over 3 pi r cubed. This is a lesson in plugging in, my friends. So I found the volume, it's 1. What was the other thing I need to know? Surface area of a sphere, which is 4 pi r squared. Oh my gosh, someone's wearing really strong perfume and my pregnancy self wants to vomit. 4, so this is area, 4 pi r squared. Surface area of a sphere. Okay, good so far? We found the equations. So, now we have to figure out what to do. Well, this should be relatively easy for you because we are given radius and we don't even have to divide. So, what happens if you were given diameter? Half it. So what would the diameter be for this one? 10. You want to be careful. They can do stuff like this to you. You take that diameter, you cut that puppy in half so that it fits in the equation. You don't sit there sweating and crying and saying, why would they give me diameter when they gave me an equation that says radius? Just divide it in half. Okay? So now we're going to plug 4 over 3 times pi times 5 to the third. And I'll find out some type of number. And then, for area, you're going to get 4 times pi times 5 squared. And you're going to get some type of number. What's the volume number, humans? What is it? 5? No.
523.
Thank you. 523.60 is what I have, yeah?
Yeah.
Okay, 523.60. And then, what is this one?
314.
Thank you. 314.16. But it doesn't want these numbers like this. It wants the ratio, specifically surface area to volume. So I'm going to put 314.16 over 523.60, and I'm going to divide that, and I'm going to get a number, and that number is going to be? 0.6. Yes, it is. It would be 0.6, but the tricky thing is, it wants me to put it in to the nearest 100. It's mean. It's mean. What are you going to put in? 0.60. Now what I would do, because I'm psychopath, retentive, I would even say, I want to make sure you know that I know what's going on here. I'm going to say, oh, I can't even. Doesn't matter. 0.60. 0.60. Now if you wanted to shovel it down, you could even have said 0.60 to make it even. Because it doesn't matter where you label it as long as these numbers are plugged in. So I could have written it as this, 0.60, and I would have still gotten the correct answer. Okay, the grid is for both bubbles, both ways to bubble. All right, now we're going to pull out this one, and then I'm going to do some ones that you're like, I don't think I'm going to know how to do that. Part of the reason I want to do these grading questions for another 11 minutes is because I want to relieve the anxiety that you have related to math and science. And some of this is like kind of scary when you first read it. Let's look at this for instance. The molar concentration of a sugar solution in an open beaker. Hey, sugar, is it ionized? No. Does it dissociate in water?
No.
No. So things like that, it should be in your noggin. In an open beaker. Hey, it's open. Right away?
No pressure.
No pressure. That sounds funny. Is 0.3 molar. Calculate the solute potential. I don't know how to calculate solute potential. Oh, dang. Solute potential of a solution. It's on the sheet. How you feeling? Better? Okay, so we're going to say what a potential of a solute equals negative I CRT. You know what else is beautiful about this? All of the variables are defined. Everything is defined for you. So let's look at I. Make sure you carry that negative, by the way, guys. Ionization constant. This is 1 for sucrose because sucrose does not ionize in water. Hey, sugar. What's sugar like? Sucrose. Ose ends in an ose. What happens if this were salt? Does salt ionize in water? Yes. Instead of using 1, what would we be using? We'd be using 2 because it breaks in half. Okay, so salt, Na what? NaCl. In water, it breaks up, right? Hello?
Yeah.
Disassociates in water. So for the most part, if they give you a sugar solution, then you're good. All you have to do is follow these directions. Da 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 da. This is 1. Fine, 1, sugar. But I'm carrying my negative with me. Then C is the molar concentration. I wonder where I find that. Oh dang, molar concentration. 0.3. Then R, what is that? Ooh, it's the pressure constant. It's probably some crazy decimal, like 0.0831. Do I have to memorize that? Heck no. It's right here. Pressure constant, yo. Then I'm going to say T. Temperature is in Kelvin, but they gave it to me in Celsius. Hey, look. Degrees Celsius plus 273 is how to calculate Kelvin. So what's 27 plus 273?
300.
Isn't that nice? 300. We're going to plug this puppy in. We're going to carry with it our negative. And what are we going to get?
7.5.
479. But no. 7.5. Please, 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 baby geniuses, follow the directions. If you went 7.48, you'd be so pissed because you're not following the directions. Bam, it's wrong. Negative 7.5. I'm going to bubble that in. My 7, I'm going to bubble that negative 0.5. Okay? All right. Are you ready to fly on your own or no?
Yes.
Yes? Okay. So, here's what I would like you to do. I would like you to do number 4, and I would like you to do number 5, and I would like you to do number 6. 4, 5, 6. This is your quiz. 4, 5, and 6 is your quiz. You have 7 minutes. /6. I'll tell you 7 and then I'll really cut you off at 5. It wasn't a chirp. Use your formula sheets. Seeing a lot of correct answers. Wrap up our answers, please, in the interest of time. These questions should have been a little bit like review, right? Some Hardy Weinberg, some water potential. All right, let's do it. Let's do it to it. Number 4, biologists are trying to determine, would you like to review these? Yes? Okay. Biologists are trying to, we'll do it really fast, okay? Biologists are trying to determine the conditions under which a desert plant can survive. Pointless information, I don't care about any of that. As part of the research, they determined that the, solute potential, talking, of the plant cells is negative 0.6 megapascals. And the internal pressure potential is negative 1 megapascals. What is the, easy, what's my equation? Water potential equals
Pressure potential
pressure potential plus
solute potential.
solute potential. Did they give me the pressure? Yes, what was it?
-1.
Negative 1 plus, did they give me the solute? Negative 0.6, which is going to give me negative
1.6.
1.6. I want to round to the nearest hundredth.
Negative 1.60.
Negative 0.60. Negative negative 1.60. Raise your hand if you got that right. Raise your hand if you put the 0 in. Okay. I'll take it. I trust you. Oh, by the way, this is also scary. Look, I got the right answer. I did it the right way, but I forgot to bubble. Sad. Fail. Negative 1.60. Pass. Okay, moving on. A naturalist working on Isabella Island observes that 24, oh no, Hardy Weinberg. And I'm giving, can you hear me? Oh, can you hear me better now? Can you hear me better now? Hear me better now? Okay. Oh, I'm not yelling, it's so much better. Okay, observe that 24 nesting birds show a rare recessive condition that affects beak formation. That's rare. The remaining 63 birds in this population show no beak defect. They're normal heads. If this population is in Hardy Weinberg, change? No change. What is the frequency of the dominant allele? So I know some stuff.
Wait.
I know some stuff. What's the stuff I know about this guy?
Recessive.
This wants to be q squared. It's not there yet, but it's going to be. How about this over here?
P squared.
P squared plus 2pq. Yes, it is. P squared plus 2pq. Pissed. People are pissed. Okay. Now, the frequency of the dominant allele, what do I really care about finding? I want to find P. So, what's the easiest route to get there? What's something I know for sure? Q squared. That's what I'm gonna use. So first let's talk about this number. This number is a bit arbitrary. I don't care about numbers, I care about percentages or decimal points. So I need to turn this into something that matters. How I'm gonna do that is I'm gonna say 24 over 24 plus 63. Or 24 over, is that 87? I wanna say yes. And that equals 0.72. No? 0.27. That's
No, I think I got it.
0.27? 0.26 or 0.27? 0.27. Okay. 0.275? Are we happy with 0.27?
Yeah.
Okay, thank you. And this is now my true what? This is my true q squared. Do I care about q squared? No, but I can get something from it. What can I get? Q. I'm going to square root that puppy, and I'm going to say q equals, what's the square root of 0.27? Because you have a calculator. 0.52. Right? That's my q. Okay, I have q. I don't really care about q. But I do know that p plus q equals 1. And then if that's the case, I know that p equals 1 minus 0.52, which means I know that p equals 0.48. Round to the nearest hundredth. Oh, I already did that, College Board. So I'm going to say, I'm going to even say 0 if I wanted to. What? Did I do something wrong? Should keep you rounded to what?
2.53.
Oh,
0.52. Because it goes 5 to 9.
0.519 something?
It goes 5 to 5.
Okay, so here's something that's going to ease your troubles. Right now you're like, what if I round inappropriately? Anxiety, eczema, hair falling out. Relax. There's a range of acceptable answers. So they're gonna give that to you. So what do we want this to be? Is this not right? So it could have been 0.53, right? And then this could have been 0.47. They're gonna give that to you. Fine. Can't do 0. I don't know why. Because they're dumb. Okay, so let's move to the next one. Here we go, 6th grade. Hydrogen peroxide is broken down to water and oxygen by the enzyme hydrogen peroxidase. I know that because it ends in an ACE, and I'm smart. The following data were collected over a span of 5 minutes. What is the rate? Oop, rate. If I had a graph, I'd call it the slope. I'd also call it the rise over the run. Of activity from 2 to 4 minutes. Well, right there, it gave me that data, and this is my test to write on. I'm going to circle my 2, and I'm going to circle my 4. So I know. I might even circle this value and circle this value. I might just go crazy with my circling. Then, I'm going to say, it's going to say round to the nearest 100. But maybe I don't know about rate. Maybe I don't. And if I don't know anything about rate, I can find something like this that says rate. Do you see it? And it says dy over dt. Well, maybe you're like, what the heck? What is dy? Oh, the amount of change. And then t probably has something to do with time. Another way of saying this is y over x, right? Or the change over x, rise over run. So let's figure this out. Well, if we were going to figure this out, we're going to use our old or our later value first. We have to calculate 2 things here. We have to say 5.5 minus what?
3.6.
3.6 over time, what? 4 minus 2. That one's an easy one to do. So I'm going to get 1.9, I think, over 2, and then I'm really going to get 0.95. Am I in the appropriate rounding?
Yeah.
Yeah, there it is. So 0.95, 0.95. How are you guys feeling? Feeling okay about this so far? Just know, look at this amazing safety net. Here's what I don't want though, you wasting all your time on here. So you got to be really knowledgeable about what this thing looks like. That's why I gave it to you. All right, we're going to do a couple more. I'm sweating. I am going. Okay, don't go away. Don't go away. Here we go. This one is one that may or may not have given you anxiety. So let's make it not give you anxiety. Calculate the probability. Oh wait, I don't want to do that one. Too easy. Let's do this one. Because this one maybe would give me anxiety. This one's easy. Calculating probability. This one's easy. Calculating rate of growth or whatever. Let's do this. Because look, annual primary productivity. Scary. Dun 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 / not. The net annual primary productivity of a particular wetland ecosystem is found to be 800 kilocalories per meter squared. Okay, that number is probably going to be important. If the respiration, so much talky, if the respiration by the aquatic producers is 12,000 kilocalories per meter squared per year, what is the gross annual primary productivity? How am I going to know that? Oh look, the equation. And just in case I didn't know that that was the equation, I thought it was just a series of letters that were maybe written on my paper for absolutely no reason whatsoever. I can go like this and say, primary productivity calculation. And that's where it is, right there. Primary productivity calculation. So, let's figure this out. Round to the nearest whole number here. Well, NPP, what is that? Net primary productivity. Did they give that to me?
Yeah.
Yes. What is it?
8,000.
8,000. GPP? No. I don't know what that is, so I'm making it an X. What about R? I wonder what the oh dang. 12,000. What do I do?
Add 12,000.
Add 12,000, solve for X. 20,000. Round to the nearest whole number. Is that tough? Oh, no. It's not, because it's already in the whole number. Okay? All right, I'm moving on. Number 10. It should be enough.
There are.
Yeah, there's no commas. No, there's no such thing as a comma. Okay? Commas are fake. Like English. Just kidding. Okay. So, 10. I'm kidding. I love you, Miss Owen. Okay, number 10. Data taken. I care about this one. I care about it. Data taken to determine the effective temperature and the rate of respiration. Oh dang, I'm running really short on time. In a goldfish is given in the data table below. Calculate the temperature coefficient Q10 for this data. Well, Q10 is something. Basically, this tells me if my degrees Celsius increases by 10 degrees, I'm going to multiply my rate by 2. Okay, so this is something like the leaf cuttings. Remember the leaf cutting situation? Except we use a solvent instead of a different varying temperature. But it doesn't matter because we're given our data here and we don't have to manufacture an experiment. But I was given Q10, and that looks like something weird that I may be able to find here, like the temperature coefficient. There it is, Q10. So I'm going to write that down. I'm going to say Q10 equals all that crazy stuff. Well, what is K2? What do you think that is?
Reaction rate.
That's my reaction rate, but it's 2.
Temperature 2.
That's my 2 though. K2. Okay, so 2. So it must be my later one, right? There it is.
So 22.
So it's 22, and then it's over what? K1, so it's over 16, and then it's all up above. What's the next thing I have to do? 10. That's a constant. So to the power of 10 over 21 minus 16, like this. See this? Literally all I'm doing is I'm plugging in values. So I took this equation right here, the temperature coefficient. Let me zoom it in for you. So I took this temperature, whoa, this thing zooms fast. Here's my Q10, K2 over K1, and then even if you're like, oh, what is K? Oh my gosh. Right there. Reaction rate, okay? And then at the second time, and then reaction rate at my first time. So I'm doing that, and then I'm having my 10, then I'm having my 10, and then this is to the power of, so zoomed, hold on. Hopefully you can figure this out. So now, what is this answer? What am I gonna get? Wait, so, oh, did I hear 1.89? No, I didn't. So I'm going to get 2, and 2 would be my answer. There it is. This is a mean one. This one, they were mean. So you're obviously, multiple people are going to get 1.89, right? Wasn't the first number you wrote down 1.89? And then you're like, 1.89, I got it. I'm a brilliant human. You are, but you're wrong. So you need to make sure you answer it in a whole number, so you're going to round that puppy up to 2. Done. I'm skipping 11 about grasshoppers in Madagascar, although I love them. Let's look at number 12. This is another one. I'm using kind of a variety of the different equations so that you can navigate through this, just so you know. That's why I'm jumping around a little bit. Ben, he has a 2 molar sucrose solution. Thanks, Ben. He dilutes it. Oh, dilutes. That's probably going to be an important word. To create 3 liters of a 1 molar sucrose solution. He's diluting into 3 liters, 1 molar. Got it. All these numbers are probably going to matter. How much of the original solution did he dilute? So we have a volume here. We don't have a volume here. We have a concentration. We have a concentration. Round to the nearest tenth. Well, I'm really looking for dilution, like that says dilution. Used to create a dilute solution from a concentrated stock solution. CI-VI, CF, VF. CI-VI equals CF, VF. So what looks scary maybe isn't. Do I have my concentration of my initial?
Yeah.
Do I know that? It's 2. Do I have my volume? X. Do I have my concentration? 1. Do I have my volume? 3. That's 1 times 3. 3. And then I'm going to divide that by 2. What am I going to get? X equal to be?
1.5.
and I want it to the 10th. Is it to the 10th?
Yes.
It certainly is. 1.5. 1.5. Would this have been scary? Okay, is it scary now? No. Plug it in, plug it in. Last one I think I want to do. I am late tired. Let's do number 13 because it has to do with pH. And we love pH. What is the hydrogen ion concentration? I got Miss G to help me on this one. What is the hydrogen ion concentration of a solution of pH 2? Round to the nearest tenth. Well, I'm looking at pH. So I want to find about, oh gosh, here it is. pH. pH equals negative log base 10 of our hydrogen ion concentration. So that's it. I literally just pulled that from here. I didn't pull it out of anything else. But one of the things that you need to know is how to take inverse logs, right? That's something that should be in your knowledge base. Let's do this first. Do we have any info that we could plug this into, that we could plug into this? Yeah, we know the pH is 2. So if we know the pH is 2, the first thing we want to do is we want to multiply, or we want to get rid of this negative, right? We want to just leave it with the log. So we're going to make this negative 2 equals log base 10 hydrogen concentration. Then I'm going to take this 10 and I'm going to put it over here. I'm going to say 10 to the negative 2 equals the hydrogen ion concentration. Because this is, everything that you will ever do, by the way, will have log base 10. Nothing in this class is that advanced, where you have to change the base of your log. You also have a log button on your 4-function calculator, which is nice. But you don't have an inverse log. I searched all over for it, and so did Miss G. So you have to know that if you want to take the inverse log of something, you know you need to bring your 10 over to here, bring your base over to here, and then raise it to the power of the pH. Now they could have asked this question differently. Oh wait, should we finish it? Okay, so what is this? Yeah, so the answer then would be 0.01 molar. And it wants it in the hundredths, so I did that. 0.01. 0.01. Okay, sorry, let me finish my thought really quickly on this one. So, related to this, they could have given us a different question here. They could have given us something easier, right? So they could have given us the hydrogen ion concentration and asked us to find the what? pH. In that case, what do you do, calculator heads?
Plug it in.
Plug it in. Plug it into your calculator. And you get your pH. That's 2. So if they would have given us that the hydrogen ion concentration was 0.01, then we're great. All right. In terms of grid-ins, by the way, these are really representative of things that you will see. And I think had we not gone over them, they may have caused you a bit of like anxiety and stress during the test. But they shouldn't now, and you should pass your knowledge on to other people. Based on a yes, I feel good, a medium, or let's do this. 1, no, 2, 3, yes, I got it, and I feel a lot more confident. There, I want to see your numbers. You can hold them at your chest if you're embarrassed. Yes. Okay, good. Let's move on.
You're about half right now. Oh.
Hey, don't let me lose you. I'll make Doris come out. Okay. Math review. Do we know about chi-square? We're comfortable. Can we do Hardy-Weinberg equilibrium? No matter what we're given, right? If I give you a P, if I give you a Q, you're going to mind them. And you're going to plug them into your equations. Water potential. You know that easy equation, water potential equals control of the volume plus potential of the pressure. If it's in an open container, no matter what, 0 pressure, no pressure. So they may or may not give you what the pressure is. But if it's in an open container, it's going to be 0. I didn't go over this, but that's okay, right? Do you know about Punnett squares? Do you know about dihybrid crosses?
Yeah.
Foil? Okay. Graded questions. We did it. How did you feel on the quiz graded questions? You're good on the 3 that you did. Awesome. Let's look at some graphing practice. Okay. I'm going to give you just a really quick PowerPoint of information, and then we'll do some samples together. So we're going to skip a lot of these slides, but I generally just want you to know what can be thrown at you in terms of graphing. This also was put on by College Board, so I wanted you to show that it's a valid thing for us to study, that it's not a waste of our time. We're going to kind of power through, but that I want you to know the different graphs and when to use them. I'm skipping over science practices and effective graphs. This is important. What kind of curve is this? It's a normal curve. It can be used in a variety of situations, one of which I went over today with my freshmen. We were talking about modes of speciation or modes of dispersion. Do you remember if we have a test and everyone pretty much in a normal curve gets a what?
C.
Everyone gets about a C, but then they did this with the MCAT. Everyone did really well. Gosh, amazing. Did they say people studied so hard and they're so smart? They said the test is too easy. So what did they do? They moved the normal curve in one direction. So they made it directional selection, essentially. If your curve is moving in one direction, you're talking about directional selection. Birth weights are an example of stabilizing selection. What do you want your baby to be? Very what? Very normal or very average. Skinny babies, no good. Fat babies, no good. Well, I mean, some are pretty good. But generally, healthy birth weights in the center. This is stabilizing selection when it's really good to be very what? To be very average. And then do you remember the last one? Disruptive selection. That's the one where the selective pressure comes right down on that normal individual, and now it selects for those two extremes. We had an example here with mice. We said that there's like light-colored mice, tan-colored mice, and black mice. And the tan-colored mice were selected against, maybe because there was a fire, so we had ash, like white and black soot and such. So these guys ended up being selected upon because they were found out. But then the white and the black mice did really well. What could this lead to over time? Yes, speciation. So if they keep not mating with each other, is that a double negative? I don't know. If they keep not mating with each other, then it could lead to two different species that will speciate. So this is where maybe a normal curve would come into effect. All right. You should consider the type of question that's being asked before you consider the data that you're going to be looking at. A lot of the questions will be talking about a total population and a target population. What's the best way to select a population? Randomly. Is randomly like, hey, Sasha, are you available to do a survey? No, that is not a random selection. What you want instead is to actually randomize and assign everybody a number. You've probably done this if you're in statistics. So if you're asked to design an experiment that talks about a target population, what you want to do is the number one thing, make sure you randomly select your population out of your total population or your target population, whether it's an age group or people that have this particular type of cancer, or whatever the prompt asks you to talk about. Descriptive statistics. You guys are going to see a lot of graphs and tables. Some of them look overwhelming and they'll come with a paragraph or 3 prompts ahead of it. However, with that being said, when we looked at some of the sample problems in the past, some can be as easy as, what's the highest percentage concentration that's present in this? And you literally go like, 2%, and you fill that in. So don't skip over the ones that look overwhelming. You want to read the question. Sometimes you want to go back. It depends on your availability or your ability of reading comprehension. Some of you can speed read and scan through all of that really quickly. Some of you read the question, see what it's asking, go back, look for it, and then find your answer in the multiple choice section. Remember, there are only 4 answer choices instead of 5. So you're going to see distributions that look like this, graphical displays of how populations will work. There are 2 types of descriptive statistics you'll see, comparative and association. What are we doing when we're comparing? You need to know these words. Yeah, you're saying, look at this population and look at this population. How are they what and how are they what? How are they similar and how are they different? So whenever they ask you to compare these 2 populations, you want to do that effectively. If you just say, they're similar because of this, bam, move on. Well, how are they different? Go back, you just lost points. Association statistics look for correlations between phenomena. Maybe it's a statistic based on how many antelope were consumed by the tigers in the valley of the desert. And in 1994 and in 1998. And how did they compare? Well, you're going to go like 94, bam, 98, bam. It's higher in 98. And that's your answer. Okay? So if it's asking you to compare those two, make sure you're looking at the dates. It's relatively easy to look at. Here's another way to compare. They love using pie charts as compare because we haven't seen a pie chart in a while, right? We don't really talk about pie charts other than the percentage of genes that are coding in our genome, which is what percent?
1.5.
1.5. We have bar graphs, we have box and whisker plots, and we have pie charts. So these are comparing variables within investigations. And then here's associations. What are these? Scatter plots. They'll never ask you to do a mosaic chart. If they do, you can come and kick me in the knee. If they're like, construct a mosaic chart on the following data set. Right here in the cap. Okay, 3 types of data. You have normal data, ordinal data, nominal or count data. So here, this is the 3 types and how it will fit. Normal data, data that fit in a normal curve. Skin colors, heights, test scores. Feel me? Okay. Ordinal data, data that do not fit in a normal curve, like amount of freckles on human X versus amount of freckles on human Y. That's counting. What kind of graph? Kind of graph for counting. Bar. Good job. And then count data is also generated by counting. It would also be your bar, your statistical analysis, or your pie chart. All right, I'm going to skip. Okay, 5 types of graphs that you could do. A scatter plot, a bar graph, box and whisker, a histogram, line graph. What's the main difference between a histogram and a bar graph? There's no space. Things touch. That's the main difference between histograms and bar graphs. So why would we want to do a scatter plot? Now a scatter plot is going to ask you to fill in a best fit line. I would like you to read through it because if you're asked to graph something, you should know what an independent and a dependent variable is. Remember, you get points for stuff like this. So if you're asked to graph something, just think, thank you, College Board. You gave me an opportunity to earn a ton of points, like by putting a freaking title on my graph, and or lose a ton of points if you don't put a title on your graph. So, give your graph a title, make sure where your independent variable is, that it's on the x-axis, and your dependent variable is on your y-axis. Make sure your scale is appropriate. I was just grading a ton of freshman labs that made me want to rip out my freaking eyelashes. And one of the things that they do is they start their side of the graph with, like, let's say 20, and then they go up by 5s. Can you start a graph with 20 and then go up by 5s? No, unless you put a break in it. So if you're going to put a break in your graph, hopefully you know what I mean by that. So let's say this is your graph, and you go like this. Then you can start by 20, and you can go up by 5s. 25, 30, 35. Also, do you want to number your life away? Probably not. So think about scales, or think about where would be appropriate, what would be an appropriate scale for the piece of paper that you're given. Okay? You want to make sure that you label both of your axes. You want to make sure that your units are appropriate. If it's telling you it's in moles, make sure you say blah, 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 in parentheses, in moles. Because you can earn points like that. You can get points removed from that. Make sure that, this sounds silly, but I swear it's part of it. Make sure when you do a pie chart that all of your pie adds up to 100%. I have seen pie charts that don't. It's like magic. The other part was just somehow placed away in a different land. Comparing different things. Okay, so look at the back. Look at the next part. The following graph is a fair to good example. Fill in the table with what's good about the graph. So this is fair to good. Crimes and a robbery. Well, we have the year. That's good. What else do we have? We have a key. Keys are good, especially if you're doing things that are in different, like you're going to create a line graph and one's going to be like that, and one's going to be like that, and one's going to be like that. You want to make sure you have a key so people aren't like, the dotted line data set represents dotted lines everywhere. You want to make sure you know what dotted line key is. It has a title that makes me happy. How about the scale?
No.
No. I don't like the scale. Why?
There's no break.
Starts with 35 and there's no break. What they needed to do is either start lower or make a larger windowed scale. Look what they're going up by. They're going up by 1. Did we talk about the thing that was published about SAT data and height? We talked about that in here? They like to do this a lot. One of the things that was published was something related to SAT score and height, and it had a kind of curve like this. What does this automatically tell you? Tall people
do better.
Do better. Actually, it looks a little bit more like this.
Denim.
Tall people do better on the SAT. But guess who was polled? Babies. Babies were polled. And they didn't do so well on the SAT. So this is up to like 3 feet. And then everyone else did pretty much the same. Just kidding. Okay, so there's that. Wait, I didn't mean to say flip. Okay, let's look at experiment 1. Okay, I'm going to talk about this for 5 more minutes and then I'm going to shut my mouth about it and we're going to move on to a new chapter.
I'm going with Nikki.
I'm doing kind of well. Okay, experiment 1. Here we go. Use the following data to create an appropriate graph and answer the questions. Graph paper on the next page. So let's look at this stuff that we have. We have depth in meters. We have bubbles per minute in plant A and bubbles per minute in plant B. So we're going to first answer some questions. What is the dependent variable?
Minute. The number of bubbles.
Where are we? Did we answer
What's
the
that? Huh? Right? What's my independent variable?
Time in minutes. Darn.
Why?
Because it doesn't depend on.
Because it doesn't matter, right? Because it doesn't change. If I dig down deeper, does the depth change? Yeah, it's going to go down as long as I keep digging. So what type of graph would be the best fit for this?
This
What do you
battery's
want
out.
to
Give me the other battery.
do? Line.
You put the wrong one in.
Let's
No, I
line
put the
it
right
up.
one in. Where's the other one?
Okay, so I'm going to set this up. First, I'm going to say, what is this going to go there? This is going to be my depth. And then this is going to be bubbles per minute. Okay? I don't need to answer that. It's down there with a happy face. What did you do for the legend? Okay, so now I'm going to have some kind of a legend, or I'm going to have a key. And the reason I'm going to have a key is because I have how many data sets? I have 2 data sets. So maybe you want a key representing one with a straight line. That's going to be plant what? A, and then maybe the next one is going to have a dotted line, and that's going to be plant B. I'm going to key that up. It doesn't even really need to look nice. I know that that's weird to hear me say with my non-straight edge up here that makes me a little bit crazy / OCD, but I would rather you focus on getting the information points down than making the box around the key perfectly square. You feel me? Okay, then I'm going to get my data. Well, let's look at the data set. Where am I beginning? What's the lowest data set? 10 is my lowest. What's my highest? 50 is my highest. And I have a pretty big piece of paper. Now, one of the things that I don't want to do is I don't want to have a graph right here. Look at it, little. See how little that would be? Little is bad. I want to be able to see stuff. I want to see it. So, really, could I start at 0? Yeah, and that makes me happy too. No breaks. And I'm not going up to like 1,047, so that's good. So I'm going to start at 0. Let's do bubbles per minute.
47.
We're doing just that first, by the way. So bubbles per minute, this is my 0. And then I get to go all the way up to 50. So how many lines could I essentially skip? Like 5 or 4, huh? So, no, that's not going to work. Oh, if I go by 10s? You want me to go by 10s?
Yes.
Okay. So 10, 20, 30, 40, 50. I might be off. Don't judge. So this is the number of bubbles per minute. I've taken up a good amount of real estate here. Are you happy with it?
Yeah.
I am. Then we look at depth in meters. What's our lowest?
2.
2. What's our highest?
30.
It's nice because as we go down, we get deeper, and so the numbers just progress. 2 to 30. What can I go by?
1.
Oh, ones. Well, ones I'll be here till 2027.
5.
I can go by 5s, and then I can skip around, right? So 5, and then how many can I skip? Like maybe 5? Okay, so 5, 10, 15. Did I do this right?
Yeah.
Ooh, look it. It's perfect. Just how I like things. Okay. Then I'm going to plot my data. I'm going to get a trend. I'm going to be able to answer questions about this. How are you feeling? Good? Okay. Yes, sir. Depth in meters. Now, what else do I need to make sure I do? Anything else? How about I need to make sure I follow my key. That's something that's important. I talked to one of my friends that's a reader. She said a lot of people mix up their keys. So they will key something first, and then they'll draw on their graph, and then their key won't match what their graph draws. Bam, wrong. Wrong data represented. Points off. Or sometimes an incredible loss of points, like wrong data represented, graph inconsistent with data. No points. So my suggestion is you start drawing out your graph, and then you decide what you want your key to be. Okay? So you're not like, what did I code A for? You can code it as you go. Cool? All right, 24. Early life, chapter 24.
Let's do it. Maddie, this is a good time. Just Maddie, oh, I could have paused it, whatever.
Hey guys, I just got a good question that was asked by Emma. She said, when we start doing our short answer, will we still have our formula sheets? I think it's a no. Because your grid-in questions happen when your multiple choice happens.
Right.
And that's when you get that perforated page with your formula sheet. Then you leave and you go to a break and you come back and like talk to me and tell me stuff. My freshmen are nervous of when you're around. Like, what are they saying? They're so smart. bio. And then
No battery in there.
you leave.
No, there is.
Then when you go back into the room, you have a whole other set of stuff. They've taken away your multiple choice stuff. So I believe you do not get your formula sheet in your short answer.
How long will it last?
Okay.
What?
Is it almost ready? Almost dead?
No, it's got like 1/4 left.
Okay. Early life and diversification of prokaryotes. Pro, old or new? Pro, old, new, Who's a eukaryote in here?
Oh, yeah.
We are. Everyone that didn't raise their hand is a freaking cyborg. So, the Earth formed 4.6 billion years ago. Not 6,000 like some people thought. Pre-who? Pre-Darwin. The oldest fossil dates back to about 3.5 billion years ago. I want you to know that they are single-celled organisms and they have no membrane-bound nucleus. Yes. They live in dense areas. So the main thing that I want to talk about, ooh, pretty. The main thing that I want to talk about is that they're very incredibly abundant on Earth. And they're almost everywhere. Places that are too acidic, or too salty, or too hot, or too cold. Places like hydrothermal vents, or at the base of volcanoes, and stuff like that. But I do care that you know this. The chemical and physical processes of early Earth. So this is really the main idea of how life came to be. And that's what this chapter kind of orients around. If you look, the two things that I care about you starting are on page 459. This list of 4 things, and then synthesis of organic compounds, the Stanley Miller experiment. Those are pretty much the only 2 things we're going to cover from chapter 24. The rest of it you already know with binary fission, or the reproduction of bacteria cells that produce clones of themselves with the exception of mutation. And then you probably are never going to be asked about halophiles or thermophiles or extremophiles. The only way that you actually will be asked, that's on page 471, is in a cladogram or in a phylogenetic tree, and then you have to associate them with the most nearest relative, or what is the one trait that made this thing different, that put it on the phylogenetic tree in this particular way. And then in that case, you're reading claudograms. And let me just look. The rest is done. So let's just look at this. Chemical and physical processes on early Earth may have produced very simple cells through a sequence of stages. So these are the stages that we're talking about. What is abiotic?
Not living.
Non-living. Pretty much throw an A before something and it means not that thing, right? Can you think of the opposite, like the lit zone of the ocean and then the not lit?
The photic and
The photic and the aphotic. So number one, abiotic synthesis of small organic molecules such as what? What's an organic molecule?
Carbon.
Such as something that encases carbon. Joining of these small molecules into macromolecules. Macromolecules, mac means big. This should automatically flash you back to what the macromolecules are. Like, what are they?
Carbohydrates.
Carbohydrates.
Protein.
Carbohydrates have a monomer. Monomer is?
Monosaccharide.
Monosaccharide, or a simple sugar. Sorry. What's another macromolecule?
Protein.
Lipids. Monomer of a lipid? Fatty acid. Not a fat. A fat is a triglycerol. It would be a fatty acid, just a component of a fat. What's the next one? Nucleic acid. Nucleic acid could be what or what? DNA or RNA. What's the monomer of that? Nucleotide. There are 3 parts to a nucleotide, eh? Sugar, phosphate, nitrogenous base. What's the last macromolecule?
Protein.
Protein. Super important. The building blocks of proteins are amino acids. Awesome. So we know that this abiotic synthesis, this non-life synthesis, production of organic molecules, which together combine to make these macromolecules that start to build the framework of life. Then the packaging of these molecules into protocells. What's proto mean? Like prototype?
Like early.
Like pre, right? Like the not the type, but the prototype, the thing that's going to lead to the type. So, membrane-bound droplets that maintain a consistent internal chemistry. Maintaining consistent internal chemistry is called something.
Homeostasis.
Homeostasis. It means you can take something in, you can put something out, you can maintain osmotic balance. You might also be able to maintain your temperature gradient in conditions that may not be favorable. And then the origin of self-replicating molecules. Self-replicating means you can take the information that is yourself and you can produce it again. What kind of sex is that? Asexual. What are you producing?
Identical clone.
Exactly the same thing. So in terms of diversity, well, what do we know about diversity? It's always good. Diversity is always good. So in terms of diversity, this is lacking, right? But it's just talking about the beginnings of life on Earth. This is producing not diverse organisms, but they have the ability to reproduce. They have the ability to reproduce asexually. What's the number one source of evolution? Mutation. That's all they really have to do. As long as they can mutate, their DNA changes. If their DNA changes, most likely their RNA changes. If their RNA changes enough, what really changes? Protein changes. When you get a protein change, you change expression, and then expression causes you to be able to what? Darwin. Evolve. Natural selection. If you're too busy to be here and you'd rather text, please leave. Okay. Next thing. Earth's early atmosphere was likely to contain water, vapor, and chemicals. This is an important component. The water vapor, the presence of water. This is why in our space exploration, what's the number one thing we're looking for all over these planets? We're looking for the presence of water and presence of possibly past life. And chemicals released by volcanic eruptions. What is released by volcanic eruptions? It's a part of a cycle. Carbon cycle. So part of the carbon cycle is released. Specifically, look at these. Nitrogen is a component, can be a component, but carbon dioxide and carbon makes us what?
Organic.
Living, and what kind of organisms? Organic organisms. We're carbon-based organic organisms. Methane is also huge, ammonia and hydrogen. So this is the building blocks of life. As Earth cooled, water vapor condensed into oceans and most of the hydrogen escaped into space. What were we left with?
Oxygen.
Oxygen or air, breathable air. Then came the idea of plants that were underwater. Remember, plants that are underwater could take in carbon dioxide. They're a process that was fab. Photosynthesis, taking carbon dioxide and producing just this thing as a byproduct.
Oxygen.
More oxygen, which allowed us to evolve even further. I want you to know about this 1953 experiment. They'll probably tell you about it in a prompt, but just so you have an idea of what we're talking about. This is the Miller and Urey experiment. They conducted a lab experiment and they set up this pipette, this whole system that mimicked the possible outcome of the production of life on Earth when there was nothing living at that time. So it showed that this is the most important part. Abiotic synthesis of organic molecules. Abiotic, again, is what? Non-living, non-life. So it showed that all of these abiotic things can come together. They can produce organic molecules, which are the building blocks of life, which then can produce atmosphere. And from atmosphere, growth would be possible. That's it. Stanley Miller. Okay? That's the Miller experiment. You may want to Google the Miller experiment, or if there's, I don't think your book explains it very well. It doesn't go into that great of detail. Last year's book does, so maybe what I'll do is I'll scan last year's book and I'll put it on the website for you, and it actually shows the experiment with how it was conducted, so you can have an idea of what I mean by it. Okay? Let's go to chapter 42.
Maddie, you have 40 minutes left, so you should be good. You have 40 minutes left, so you should be good.
New rule, no perfume in the next session. Thursday, if you're invited back. Okay.
Yeah.
You don't? Well, that's okay. Ecosystems and energy. So I want to do a really quick, you know what? What chapter is cellular respiration? Is that 9?
8, I think. 7 or 8.
7.
8 is photosynthesis.
Oh, it's good.
Yeah. Wait, isn't it 8?
Okay, good. I want to do a really quick turbo review of energy and how energy works, okay?
Yeah.
Forms of energy. First of all, energy of movement.
Kinetic.
Kinetic. Energy of opportunity. Potential. Energy of consumption. Can be. Can produce heat, thermal, or movement. Hi, you're so down on the floor. And you're also digesting. So this is you after Thanksgiving, the unbuttoning of the pants. You know, you're like, whew, it's hot in here. And you just take a nap. Chemical, good. The breakdown of food. So this is the whole idea of transferring of energy. Can it be lost?
No.
No. Can it be transferred?
Yes.
Yes, energy can be transferred. So one of the things we'll talk about in terms of energy here is that energy transfer and how cycling will occur. But we have a couple of other things. If we start out really high, we have a high amount of what?
Potential energy.
Potential energy. A high amount of potential energy. The reaction exists that brings us down. What is released? Energy is released. This also takes us to bonds. If we're forming bonds, we're doing what with energy?
Consuming.
We're absorbing energy. If we're breaking bonds, we are releasing energy. And we call these different things. When we go from very high amounts of energy to very low, we're combusting and we're releasing energy. What is that called? Exergonic or spontaneous or catabolic, or change in G is? Negative. Awesome. If we have to build something, like the guy in the blue bathing suit, remember him? The guy in the blue bathing suit up on that diving board? If he has to build to go back up onto that diving board, what is he doing? He's raising his potential energy because he's putting in work. He's putting in work, he's expending energy, and he's raising his potential energy. This costs energy, but he's building. What is that called?
Endergonic.
Anabolic, or
Endergonic.
endergonic, or non-spontaneous, or change in G is
Positive.
positive. Okay, so do you recall all of this? Yes. All right, cool. So let's look at ecosystems and how this works. First, for this, you need to know levels of organization. So, levels of organization that should be committed to your noggin are biosphere, biome, ecosystem, community, population, individual cells. Do it again. Biosphere, biome, ecosystem, community, population, individual cells. When we're talking about an ecosystem, this is something that people mess up quite a bit. They mess up ecosystem, population, and community all the time. But an ecosystem isn't just the animals, it's the what?
Abiotic factors.
The living and non-living factors. So it's the rivers, it's the dirt, it's the trees, it's the microbes, it's the detritivores. Remember all those guys? So everything that kind of encompasses it. A community is just the what?
Living.
Just the living things, but any living thing. And then what's one smaller than a community?
Population.
Population, which really we could call
Species.
a species. A group of individuals that can interbreed and produce
Fertilizer.
viable, fertile offspring. Gosh, you're fab. How'd you get to be that way? Okay, so moving on, we're going to look at this 42.1. You know law of thermodynamics, you know law of energy transfer. We kind of have already gone over that, but let's just really quickly look at it. That the first law says energy can never be created nor destroyed. It can only be transferred or transformed. The second law has to do with entropy. Remember this?
Chaos.
What's another word for entropy?
Chaos.
Chaos in the universe. Is entropy a good thing?
Yes.
Yes. Entropy is a fabulous thing because it allows us opportunities for bonding. If there's breakage or if there's forming, then there's change. And change is a good thing because it's causing us to exchange energy sources. What happens if you stop exchanging energy sources? You're dead. So you're doing it right now. You're exchanging. Look at, isn't that great? All right. I'm going to move on. I thought that I had a thing related to this. Let's just do this. The sun. It's amazing. It's also the #1 source of energy on Earth. Here, it's going to light up the primary producers. Producers are guys that can make their own what? They can make their own food. They do this through a process known as, but they also do this other thing, cellular respiration, where? In their roots, in their root system. From here, we have some heat loss. Is it lost? No. It's transferred. Good. From here we have our primary consumers, and our primary consumers are going to feed on our primary producers. What amount of energy do they get? 10%. What's the level that we move up from one to the next called? Starts with a T.
Trophic.
Nice. It's a trophic level. So as we move from a primary producers at the base of our pyramid to our primary consumers, the primary consumers can consume 10% of this. Why? How come? The rest is lost as heat or needed for processes for your producers. So 90% is needed for them to sustain their own life, their own world. 10% is available for transfer. From here, our primary consumers go into our secondary or tertiary consumers. Secondary consumers a lot of times eat primary consumers, but they could also eat primary producers. Anyone in here do that kind of thing? It's so strange. It's called being a carnivore and an herbivore. Yes? What are you? You're an omnivore. Yes. So then you have a lot of this cycling that's happening. And what's happening here is chemical cycling. We have our nutrients that are transferring from one to the next. You also see this detritus. Detritus. What is this being produced? Who does this? Decomposers. Decomposers breaking down dead and organic matter. So all the poop, all the vomit, all the death. They like it. From here, we have microorganisms and other detritivores, and we're cycling this back. Notice it cycles back to here. What do primary producers like? Fertilizer. Primary producers love fertilizer. So all of the dead and dying and poop is good for them. So thanks, microorganisms, for in a sense allowing me to have a cheeseburger. Okay, we know what primary production is. Primary means what? First, here's your gross primary production. Gross just means the total amount. It doesn't take anything out. So it's not the, what's not gross, but think money. Net. Not your gross, but your net. So the GPP is measured in the conversion of your chemical energy from photosynthesis per unit time. Gross means the total primary production. Who's doing this? Producers. Can primary consumers produce? No. They can produce babies and stuff like that, but they can't produce energy. The only thing that produce energy are things called producers. Then we have our NPP. This is the equation that we just did in a grid-in question, but it kind of gives you some basis of what it means. This is our net primary production. It's the GPP minus the energy used. So the gross is everything that could possibly be produced by your producers. But then some of that is going to be used by your consumers. So the NPP is your net primary production, and it stands for your GPP, or your gross primary production, minus the energy used by your primary producers, the energy consumed, for autotrophic respiration, for respiration, essentially. What is an autotroph?
Something that makes our own food.
Yeah, it's a primary producer, right? It's a making of a product, essentially. It's expressed as this, but you'll never be asked about how it's expressed. All right, I'm kind of going to skip a little bit to, you'll never be asked that. This. Eliminating nutrients. First, let's say we have an environment. What do we know about organisms? They're going to have how many babies? A lot. And how do we define that? More than the environment can support or can sustain. It's going to continue to grow exponentially, and they're going to continue to consume those resources until they reach their what?
Carrying capacity.
Carrying capacity. Carrying capacity C, represented by the letter K. Okay? Carrying capacity is represented by the letter K. And this just means we've run out of space, or we've run out of food, or we've run out of water, or whatever that is. All of these things that we're running out of, they're limiting nutrients. So they're nutrients that will eventually run out. It's the element that must be added for production to increase in an area. So if you've reached your max, but then you have, and let's say that this organism only feeds on blackberries. Blackberries are like, that's it. They're kind of prima donnas about it. Well, once they reach it, organisms are going to start dying unless what happens? A bloom of blackberries. A huge bloom of blackberries. Then that limiting nutrient has gone up. The carrying capacity of the environment has gone up. More organisms can survive. They're obviously going to be competing no matter what. And competition is a good thing. But if they're competing for resources, or they're competing for mates and such, things like that. All right, not going to talk about stuff. Nope.
What about the other one?
No.
This
Secondary.
size. Find one this size.
Okay. Secondary. So we talked about primary production. Now we're going to talk about secondary production. It is the amount of chemical energy. This is the whole idea of additional biomass or secondary production. Here. And we do this, or we measure the production efficiency by this particular equation. If you're asked to do this on a grid-in question, what are they going to give you? They'll give you this equation, but let's look at it. It's the fraction of energy stored in food that is not used for respiration. What is respiration? What are you doing when you're respiring? You're breathing and you're breaking bonds. When you break bonds, what are you releasing?
Energy.
In the form of?
Heat.
ATP. You're releasing energy in the form of ATP. And you're doing this because you can break things down to simple molecules. What's the simplest molecule for cellular respiration? Not the simplest, but the one you're going to break. Glucose. Yes. Glucose being sugar. Into two molecules of pyruvate or pyruvic acid. Good. So we're breaking this down, we're throwing it into the mitochondria, and this is the amount that we're using. But we also have an amount that's stored. Where is it stored? In your fat. Where else is it stored? In your muscles. In your fat and in your muscles. What's the first thing to go? Fat initially, and then your muscles, And then if you have no muscles left, can you grow them back?
No.
No, impossible. That's why if you don't have any, if you've lost all of your fat and lost all your muscles, you're just like a flabby little bag of skin, right? So how does this work? Production efficiency. You have a net secondary production times 100% over the assimilation of primary production. This just means how much total production would be needed in order to be able to consume anything. So you have your primary production, which is going to be a bigger number than your secondary production, because this is all of the possible energy and biomass that's available in the entire ecosystem. Goodness. Okay, now, no. Oh, we already did this, right? Remember, look, trophic, 10%. Got 'em. But if you look here, this is what's showing you it. And you may see something like this. By May, I think you 100% will see an image that looks just like this. Or something related to how organisms will go up in the food chain. You see that our tertiary consumers consume on all of the other levels, primary consumers only on primary producers. And you see that the amount of energy that goes up, we start out with 100% of energy from the sunlight, 1 million joules. And as we go up, we have what loss?
10%.
We have a 10% loss, and that's all we're talking about here. Then we have some other pyramids. Whenever you see a pyramid on your test, you want to make sure you know what type of pyramid you're getting. Could be an energy pyramid, could be a biomass pyramid, could be a pyramid of numbers. They look all very similar, but the questions in the multiple choice are going to be geared toward what type of pyramid that you're looking at. So be sure you're answering in the right form. This thing is most ecosystems. So here we're looking at dry mass. What kind of pyramid is this? Biomass pyramid. What does it tell you about primary producers? Dang! They have a lot of biomass. They probably also produce a lot of what? They probably produce a lot of energy. Good. I'm skipping some of this. I'm skipping until we get to cycles now. I just got bored for a second. Okay. Here we go. Biogeochemical, crazy word. Bio means biology or life. Geo means of the. Chemical means having to do with molecules or chemicals. And a cycle means whatever you end with, you need to start with. And then whatever you end with, you need to start with. So living of the earth, organic or something related to molecules, and it's a cyclical nature. So we're going to look at these 4 different ones that are important. Here's the water cycle. Hopefully you know about the water cycle already. You can speak knowledgeably about it. When rain comes down upon you when you are feeling blue, it's called
Precipitation.
Precipitation. When it comes off of bodies of water or land, we call that evaporation. When it comes out of trees, we call that transpiration. When it smooches up into clouds, we call that condensation. When it comes down into the soil, we call that accumulation of groundwater. The groundwater will eventually impact and go out to the ocean. Go out to the ocean. So we have rain that's coming down. We also have runoff. Runoff is important because it can be collected in rivers and streams, and we can check to see how water is cycling. Precipitation also happens over the ocean. So this is water cycle. Easy? Got it? Okay, good. Sorry I went so turbo. Next thing is the carbon cycle. Super duper duper important. And this is kind of a simplistic version of this carbon cycle. The one in the CP Bio book is a little bit more complex. But what you need to know is that we use carbon in multiple facets. We're also made up of carbon. We have CO2 in the atmosphere. Who loves it? Plants love the CO2. So here, CO2 in the atmosphere is being taken up via photosynthesis. It's a what in photosynthesis?
Reactant.
It's a reactant. Then, what's being released?
Oxygen.
Oxygen is being released. That's not shown because we're just looking at the carbon cycle here. But we have cellular respiration that's occurring, and in cellular respiration, it's the opposite of photosynthesis. What are we taking in?
Oxygen.
CO2. What are we putting out?
Oxygen.
I did that backwards.
But yeah.
What are we taking in?
Oxygen.
What are we putting out?
CO2.
CO2. That's why we're putting CO2 back out into the atmosphere. Watch me do it. Just did it. Okay. Then we're consuming things. Look at him. Hungry. And this is going this way. Hey, this is also something that I wanted to remember. The arrow is going this way. The arrow always goes to the eater from the
Eat
tin. Because it's energy transfer. If this rabbit is dead, can it get any energy?
No.
No, it's dead. The only way it's going to get energy is by transferring it to this guy, and he's ready for it. Look at his face. He's ready. Okay? Then some other stuff is going to happen. Like, this guy is going to get eaten, but maybe some bones won't get eaten, and they're going to become a part of this, right, Adam? Still. And then this guy is going to poop after, and then his poop is going to get condensed, and it's going to smush down into decomp. Decomposition is a good thing. Eventually, this can compress over millions of years, and it can form something awesome. Fossil fuels. Fossil fuels. A renewable resource?
No.
No. A non-renewable resource. These guys are also eating, look at them. Eating, consuming. And when they consume, they're breaking glucose and they're releasing what? And CO2 into the atmosphere because they're undergoing cellular respiration. Then we have this sad state of affairs / what keeps our world going. The burning of fossil fuels. The extraction of things that took millions of years to get. And then the burning of them and the releasing of CO2 into the atmosphere. Happiness and rainbows. Any questions about this cycle? Okay, nitrogen, let's go there. Is nitrogen next? I don't know. All right, nitrogen cycling. We have 2 more images and we're done with this one. And then we might get to all the agenda items if you want me to chant cellular respiration with you. If not, I will. Okay, nitrogen cycling. So there are a couple of terms that you should know. You should know fixation or nitrification and denitrification. What are you doing when you're deing something?
Undoing.
You're taking it away, right? You're deing. I'm deing this. Denitrifying this. So we have nitrogen that's present in the atmosphere. Then we have industrial fixation, or the bringing about, the sucking in of the nitrogen, and the using of them as industrial fertilizers. Fertilizers are great. What do they make your grass?
Grow.
Not just grow,
Healthy.
they make it green. Remember we talked about the little white pellets that come in your plants? Little white pellets are little white nitrogen balls. Anyway, so this will make your fertilizer nice and beautiful, except for water is eventually going to run here, and then it's going to run it into the ocean. So there's that, but. So then we have decomp and sedimentation that occurs. We have NH4, what's that? Ammonia, right? So we have this, we have NO3, nitrate, that occurs. And we have that whole process of denitrifying bacteria or ammonifying bacteria. Do you remember that in the last chapter? So I'd flash back to your plants and know what those 2 things are. We also have this terrestrial cycling. So this has to do with more of the water and such. This is what we talked about before. Fixation in your roots. Remember, your plants can take in ammonia or they can take in nitrate, but they can't take in nitrite. So you have assimilation that's happening. Decomp, eventually he's going to die and contribute to the fertilizer. Thank you, little weasel-looking thing, weasel thing. And then we have ammonia fixation. We have uptake, which contributes to the process of photosynthesis that occurs in nitrogen. So I would know that nitrogen is a cycle, that there's multiple ways that it can be cycled throughout the atmosphere, that there's a process called nitrification and denitrification. There's also nitrogen-fixing bacteria and ammonifying bacteria that all play a role in the nitrogen cycle. Last one, but certainly not the least, this is your phosphorus cycle. Phosphorus is a major part of ATP. It's one of the most important parts. It's also a major part of nucleic what? Nucleic acids, phosphate. So we have this, phosphate, the most important inorganic form of phosphorus, which I would know. Largest reservoirs are sedimentary rock with marine origin, the soil, oceans, and organisms. Phosphate binds to these soil particles and movement is localized. So basically what you can see here is you can see leaching that's happening. You have dissolved phosphate that occurs, plankton taking it up, plankton breaking it down, plankton taking it up, plankton breaking it down. Geological uplift that occurs. Then you have dust that brings it kind of in the air. The weathering of rocks, so the breaking down of sediment, producing phosphate that happens. Runoff that carries it down the streams, making it kind of like a cloudy looking stream or water. You have consumption. Plants take this up. It aids in cellular respiration in addition to photosynthesis. And then fungus breaks it down in decomposition. So this is like multiple cycles. Although the entire phosphorus cycle is one big cycle, what I'd like you to know is that it works in tiny little subgroups with one another. Okay, I'm done with talking about this. One of the things that I didn't talk about yet, but that I want you to know about, is they could possibly ask about renewable resources, desertification, deforestation, rising water levels, lowering acidic waters, things like this, or too basic of waters. So I want you to know that you're always going to err on the side of the ecobiologist. Let me say that again. No matter what your opinion is, which I love and I value in a great discussion, but when you're writing for these essays, I want you to err on the side of what would an ecobiologist think of this, and how would they solve this problem. It should always be about less use, not wasting energy, so keeping as much energy as possible where you can, holding on to renewable resources, making sure that you live in a sustainable way. Water, you live in California, I don't know, maybe they'll have something, it's a world test, so maybe not. But anything related to sustainability is what you want to focus on. Okay, so here's my question for you. We have like 3 minutes. Are we done? You look done. So, but I swear, we actually are really, really wildly accomplished today. One of the things that I would love for you to do, before you leave me, let's just look. You have your practice FRQ. The practice FRQ serves two, hang on a minute please. The practice FRQ serves two needs. It is a 2005 AP Bio question. I gave you a piece of paper for which to write on if you need it. But then it also has to do with graphing. So it speaks to one of the themes that we covered today. And I'd like you to just do this. The answers are on the back here. I would flip it back over. Don't really look at it until you already write on it. But this is a really good opportunity for you to write. We will try to chant cellular respiration next time we're together, but we have 2 other chapters that we need to cover.